I'm Laura Harris. I'm the project leader of the GWAS Catalog. Thank you for joining us this afternoon. I'm also joined online by my colleague Maria Cerezo. Some of you may be very familiar with GWAS and working in the field already, but some of you may be new to the field. So I'm just going to first spend a bit of short time talking about the key concepts in GWAS, why we need the GWAS Catalog, and the sort of questions you can ask of it. I'm then going to go into a bit more detail about the data structure. Then I'm going to give you a hands-on look at how you can access the data through the web search, and you can play along with that at home. I'm then going to show you how to get additional data from summary statistics files, and also show you how you can submit your own data to the GWAS Catalog. So what is a genome-wide association study? So GWAS relies on the concept that for complex diseases or phenotypes, there may be multiple variants in the genome that affect the likelihood of an individual having a certain trait, along with environmental factors. The GWAS approach is a well-established hypothesis-free method that scans whole genomes for variants associated with diseases or traits. So how do you perform a GWAS? To identify variants associated with a trait, for example, a disease, individuals are genotyped at common variants across the genome using genome-wide SNP arrays. Data is then analysed to find variants or haplotypes with a higher frequency in cases, i.e. individuals with the trait, than controls, i.e. individuals without the trait. Statistical analysis is then carried out to indicate how likely the difference in variant frequency is to be associated with the trait. There are two key pieces of data from a GWAS. Firstly, the p-value, which is a calculation of the significance of the difference in frequency between cases and controls, and also the effect size, which is the magnitude of the difference between the groups. It's also important to remember that each variant in the GWAS merely tags a region of the genome because of LD. That significant variant from your GWAS is not necessarily a causal variant, and we always need to do follow-up study to establish causality and disease mechanisms. A typical GWAS study design involves two stages. A discovery stage, where anywhere between 100,000 and over 10 million variants are analysed. This population may be divided into cases and controls for a binary trait like a disease, or drawn from the general population for a continuous trait like BMI or height. This population may be homogeneous, or it may be drawn from different ancestral backgrounds, or they may have some other common background, such as they're all smokers or they're all on a particular medication. A smaller number of the most significant variants are then often followed up in an independent cohort using a directed genotyping method. At this point, they may be tested in other ancestries. And then finally, as I mentioned, the GWAS is always followed by functional studies to establish causality and investigate disease mechanisms. Which phenotypes can you study with GWAS? The GWAS is ideally designed to study complex phenotypes, by which I mean non-Mendelian phenotypes caused by multiple genes in combination with environmental and lifestyle factors. They may be diseases or quantitative traits. Often common diseases such as type 2 diabetes, but also rarer diseases such as ALS, frontotemporal dementia, or increasingly we're seeing GWAS of endophenotypes such as biomarker levels, which are then broadly applicable to a number of biological investigations or disease research. In fact, there are a huge range of traits in the GWAS Catalog. At the last count, there were over 2,500 different ones, from very specific biomarker levels right down to political preference. The GWAS approach is widely used and has achieved considerable success. Since the first one in 2005, close to 5,000 publications and 200,000 associations have been reported. So why do we need the GWAS Catalog? The aim of the GWAS Catalog is to provide summary data from published GWAS studies. At this point in time, there's no standard for reporting GWAS. There are different study designs, different ways of representing the data, different criteria for replication. Sometimes GWAS data may be the main goal of a publication, or it may be buried in supplementary table 40. There might be a single GWAS, or there might be many GWAS within a publication. So there's a wealth of data there, which is otherwise inaccessible to users without a large amount of time spending undertaking systematic review. Our job is to make sure that each GWAS is represented in a consistent way. We take published GWAS studies and reduce them to simply described associations. A particular SNP is associated with a particular trait, with a particular effect size and significance, and it was demonstrated in a particular population of individuals. We then make this freely accessible and searchable through our web interface. We're also one of the major repositories of full genome-wide summary statistics data, which we also make. So this allows investigation of all variants on the array, not just the most significant ones. And we make these available through our summary statistics repository, which can be accessed through the website. Our users come from all different backgrounds, gathering statistics on particular disease areas, using the catalogue to prioritise candidate loci, understanding disease mechanisms, and drug target discovery, to name but a few. Some commonly asked research questions that people come to the catalogue with are: what are the best candidate loci for my functional study? Have any of my significant associations been discovered previously? Are there any previously known associations that have failed to replicate in my data set? And which of my SNPs of interest have been associated with other phenotypes before? In terms of the summary statistics repository, the increased availability of summary statistics has massively expanded the utility of the GWAS catalogue in downstream analysis. So summary statistics are used in things like GWAS meta-analyses, polygenic score analysis, integration with other omics data, and Mendelian randomization, among other uses. GWAS catalogue data is also incorporated into many other tools and databases. So even if you're not using the GWAS catalogue directly, it's useful to understand where the data comes from and how we structure it, because you will see it in all these other resources. So now I'm going to spend a little bit of time talking about how we represent data in the catalogue. So we extract data following strict eligibility criteria to ensure consistency across studies. Studies must be genome-wide or targeted array-based. It's array-based genotyping, and we also accept summary statistics from whole genome sequencing studies. If it's array-based, there must be more than 100,000 SNPs assayed on the array. For each association, it must be significant less than 10 to the -5 in the overall population. So that's discovery and replication if there's a replication population. And we extract the most significant SNP at each independent locus. The key part of the key basis for the data that we include in the catalogue is the publication. Each publication may contain multiple GWAS studies, and for each study, we record separate experimental and sample metadata. In terms of the population, we record sample size and ancestry, and I'm just going to quickly talk a bit about how we represent ancestry in the catalogue. So ancestry is captured in two forms, in a free text sample description and a structured sample description. The free text allows us to represent most closely the definition used by the author, and the structured sample description uses controlled terms to increase consistency and comparability between studies and improve the search functionality. So just to give you an example of how this works, here's an article which you can see they've already told us from the title. It's a study of Finnish population. There's quite a lot of detail in the text about the family structure. And here's the GWAS catalogue entry with the discovery sample description, which I'm just going to talk about on the next slide. So here's the free text sample description, and that uses the most granular term that the authors have reported. So in this case, that's Finnish. We've also used that to explain what the case control breakdown is. So you've got 873 cases and 842 controls. And because this is a family study, we've also included some information there about the number of families. Here we have the structured sample description. So here we're telling you the total number of individuals in the analysis, and the Finnish population maps to the broad category of European. And as I say, we use those control terms to increase comparability and consistency between studies. You can see on the slide the full list of controlled terms that we use. If you're interested to read more about how we represent ancestry in the GWAS Catalog, you can have a look at the reference there from Morales et al. 2018. So moving on from the sample description, so we also provide technical information about the array and the number of SNPs analysed, and then we move on to the results. In terms of association data, we extract the variant, the effect allele of the gene, and the genomic position. We extract the strength and the effect size of the association. The effect could be OR for a case control study, or beta for a continuous trait like BMI or height. And of course, we extract information about the trait, and I'm going to talk about that a bit on the next slide. So there are two levels of trait description. Each study is assigned one or more terms from the experimental factor ontology that best matches the examined trait, and then we use a free text description, which I'll talk about in a second. So an ontology is a set of controlled terms and their relationships, kind of like a family tree of traits. So for example, in this example, you can see that estrogen negative breast cancer is a subclass of breast carcinoma, which is in turn subclass of cancer. And we could also see, if we looked for example, BRCA1 positivity, that would be linked to breast cancer within the ontology, so it also covers relationships between terms. And it also includes synonyms, so we can support synonym searching. So each study is also assigned a free text reported trait, which, similar to the ancestry, that one closely matches the information that the author has provided us. So for example, here we've annotated the study with the ontology term chronic childhood arthritis, whereas the reported trait uses the author language juvenile idiopathic arthritis, which is also listed as a synonym for chronic childhood arthritis within the ontology. And again, of course, you can see how it accounts for hierarchy, with chronic childhood arthritis is a child term of rheumatoid arthritis, which is a child term of autoimmune disease. The free text trait is also used to describe a bit more detail about the study design. So, for example, this one has been annotated with two terms from the ontology, systolic blood pressure and sodium measurement, whereas in the reported trait, you can see that it's actually an interaction study of systolic blood pressure and sodium interaction using a 2 degrees of freedom test. And that difference between the free text reported trait and the ontology term is quite important to understand when you're looking at the data that you're pulling out of a GWAS Catalog search. So I'm now going to move on to how you access the data. So I'm going to cover how you search by publication and study, how you can search by trait and use the locus-zoom plot tool, how you can search by variant and use the LD plot tool, searching by gene, accessing summary statistics, and downloading data. So I'm just going to switch my screen to the website, and hopefully you can all see that. Just maximize that. Okay, so here we are on the GWAS Catalog homepage. So by default, the search will show you the 20 most recent publications in the catalogue. This is a free text field, you can type anything in there. If I type that, you can see on the side there are 5 different types of data that it returns, and you can filter the results by clicking on those bars at the side. I'm now going to focus on the specific type of searches you might want to do. I'm going to start with publication because it's a useful way of showing you the different features of the search, even though it's probably not the most common common type of search that people do. So you can start, you can search by any word in the citation, for example, the author, but that can be a bit non-specific. So we recommend that you use the PubMed ID if you want to pull out a specific citation. So I'm just going to type in a PubMed ID there. Oh, that's the wrong one. There we go. So that's pulled out the paper that I wanted, Char et al. So if you click through, that will give you a summary of all the data associated with that publication, including links out to PubMed and Europe PMC to the citations. So this is showing you here that that there are 81 associations and 6 studies for this publication. And what that means is that there are 6 separate GWAS, each of which has its own entry in the catalogue. So if we can either click on this or scroll down to the associations table. This table is fully customizable. So you can sort by for example, if you wanted to see the most significant association, you can sort by p-value. And you can search within the table for particular things. So if you wanted to search for a particular gene, you could pull those out like that. If we then scroll down to the study table, you can see that the publication has 6 separate GWAS. This is a GWAS of facial features. If I just expand that table to show all of them. And you can click through on the study accession to the study. This gives you the sort of overview of the sample description, but you can click through to the study page and get more full details there. So on the study page, in this first box, you've got the sample and experimental details. Here you've got the sample description in free text and structured version, as I mentioned before. You've got the genotyping information. And again, the citation information. So scrolling down, we've got the list of associations for this study. And that's got links out to the other tables in the database that I'll show you later, such as the gene page, trait page, and variant page. So I'm just going to return to the search page. And now I'm going to show you how to search by trait, which is one of the most common things that people want to do with the catalogue. So say I'm interested in BMI, I can type that in here. That you can see that that's listed as a synonym for body mass index. So it'll auto suggest any traits that are relevant. So I can click on that. It shows me all the relevant traits there, hopefully with the most relevant one at the top. I can click through to that, and that will summarize all the data in the catalogue for that study. Sorry, that trait. So again, there are links out to other resources here. You can look at that trait in the ontology lookup service or ontology cross-references, or if you're interested in finding out how that's represented in other ontologies. You can look for that trait in the Open Targets drug discovery platform, and you can also look it up in our sister publication, the GWAS Catalog. So this is taking a little while to load, I'm afraid. So while that's load, oh, for that, it's telling me the page is taking too long to load. So here we've got the. If we click on this plus button here, it will show us all the reported traits that map to the ontology term. So those are the more detailed free text traits that I mentioned, like showing you that they're interaction studies, for example. It will also show you here any child traits in the ontology. So those are more specific traits that become as subtraits in the ontology. There we go, it's finally uploaded. So now I can click on that and show you. So you've got obese body mass index, overweight body mass index, a longitudinal body mass index measurement, for example, which are all child terms of BMI in the ontology. So you have the option of including those more specific terms, or you can uninclude them. I'm not going to demo that because it will take too long to load up the data. But if you see that there are those more specific terms there, but actually you're really only interested in BMI itself, you can uncheck that box and that will remove those more specific terms from the summary. So again, we've got the associations table and the study table here. One of the useful things you can do here is filter by a particular reported trait. So if you're actually looking for studies where there's an interaction with smoking, say, you could search for smoking in that box, and that will filter on those traits. You can also filter on the sample description. So, for example, if you were interested to know if there were any studies of Hispanic populations, you can search in there for the structured ancestry term, and that will filter. And if you've filtered your list and customized it in any way, you can download the output in a CSV format using that button there. Scrolling down to the bottom, we've got the locus zoom plot, which summarizes all the catalogue data from that trait of interest on the page. So you can easily see what the most associated locus is. And there you can see, obviously, it's the FTO locus, which is a very well characterized locus for BMI. And that mouse over will give you all that information to the data from the catalogue. So just going to go back to the search. So another use case you might have is wanting to find all the data for a particular variant. So if you type your variant ID in the box there, the RSID, again, click through to the variant page. Again, it will show you general information about the variant at the top. It's got links through to Ensemble, which are customized to be of most use to the types of users of the GWAS Catalog, dbSNP and UCSC Genome Browser. So basically the features of these tables are the same as on the other pages. You can link through, again, if you're looking for this variant and you think, okay, it's associated with platelet count, I want to know what other variants are associated with platelet count, you can click through and it will take you to the trait page. There's two interesting features on this page. So one is the trait table. So this is a really easy way of seeing what are the most commonly associated traits with that variant. So for example, here you can see that there are 8 different traits that have been associated with this variant. Platelet count has been associated 5 times. And the other ones less, but you can see that clearly that variant has some role in hematology traits. Scrolling down to the bottom, you've got the LD plot. So this will show you all SNPs in LD with your variant of interest, both in the GWAS Catalog and in Ensemble, and that's denoted by the different shapes on the plot. The colours on the plot show you which trait category those variants have been associated with. And you can change the reference population using this drop down at the side. And that will show you the LD for your population of interest. So there's one final type of search that you might want to do, which is searching by gene. You can type in the gene, your favorite gene in there. The annotation that we use for genes is it will be any genes in which a SNP maps or the closest upstream and downstream gene for intergenic SNPs. So again, this will show you the associations and studies for that gene, and the traits that have been most commonly associated with that gene. So one final feature I just want to show you on the search is So I mentioned we have a sister resource, the GWAS Catalog, if you're interested in polygenic score analysis. So I'm just going to type in a PubMed ID there and click through. And you can see that this one is also referenced in the GWAS Catalog. So if you're interested to find out more information about that, you can click through. That will take you to the GWAS Catalog and give you all the data that you need about the polygenic scores and how it's been calculated. So hopefully you find the web search useful. There might be times, however, when you want to do a more complicated search, or you want to search for multiple variants, and for some reason the web search is just not an appropriate way of doing it. So we do also provide the entire catalogue as downloadable flat files, if you just click through to download, and they're all there. So we've got associations, studies, and detailed ancestry data. You may also be interested in our API, so that's programmatic access to the catalogue data. All the documentation is on the website. If you're a bioinformatician or you've got a bioinformatician in your lab who can help you with that, it can be really useful to search iteratively. For example, if you've got a long list of variants that you want to pull data out of, or you want to do more complicated combinations of search terms, the API can be really useful. So I'm going to finish by this section by just showing you how to access the summary statistics. So there are two methods of accessing summary statistics. One is from the download page. So if we go to download and summary statistics, there are two tables on this page, which the loading is a little bit glitchy. I hope that's going to load up in time for me to show you. That's in the process of being fixed at the moment. We make summary statistics available from published studies, so all of the studies in the catalogue are eligible to make their summary statistics available. We also have recently started hosting summary statistics from pre-published studies, so those are in that table there. You can search within this table, for example, if you want to search for a particular trait, you can filter the table on those. Obviously, you can filter on PubMed ID and author name and so on. Or you can also go directly from the from each publication. So if where you see this little icon on the side, that means that full summary statistics are available. I'm just going to type in a PubMed ID to show you a good example. So here is a publication that has summary statistics. If we scroll down to the study table on this page, you can see that it's offering me FTP download or API access. We have a separate API for the summary statistics, which are about we're trying to add summary statistics to the API, to the database that's accessible through the API. Only about 50% of the summary statistics are available through that method at the moment, but we're adding them all the time. But all the summary statistics are available through the FTP download. So if you click on that, it will take you to the folder. You can see there are two different types. So there's the original data file, and then there's a subfolder called harmonized. And in that we make two different types of files available. So there are formatted files which have standardized column headers. So the data hasn't been manipulated in any way, but we've standardized the column headers. And there are also harmonized files in which all the genomic locations are reported to the latest build, and all the alleles orientated to the forward strand. So that can be really useful if you're trying to combine summary statistics from different sources. Okay, so I'm going to switch back to my presentation. So you can submit your data to the catalogue. We accept data associated with peer-reviewed papers. We accept unpublished data, so data that's in press or associated with a preprint or with no manuscript. Data files must conform to our standard format, so please read the documentation. There's a link from the front page. It gives you a nice wizard to help you create your submission, and you can then submit your files and your metadata in a standard format. So if you get stuck with any aspect of the GWAS Catalog, please get in touch. We have a really active Twitter feed if you're on that. We have a low volume mailing list, GWAS Announce. We have 2 help desk emails, GWAS Info, which is the main one, which we can help you with any aspect of the catalogue, and GWAS Subs, which is for support on submissions. And that's it from me. So hopefully I've got time to take some questions.
Thank you very much, Laura, for today's webinar. And thanks to all the attendees for listening to the webinar today. Just before we go to the Q&A session, I would just like to say that please visit our webinar webpage to find the list of webinars that may be of interest to you. And as I mentioned earlier, that after you close the webinar, there will be a survey that would launch. So please share your thoughts about the webinar. And with that, I will now move on to the Q&A session. So please keep typing in your questions in the chat box, and we'll soon answer those. So we have got a few questions in there. So my question is about the sample size of each available GWAS summary statistics file. One can get information on the sample size of the discovery and replication from the downloadable list, but it doesn't seem possible to know if the summary statistics were calculated using the discovery sample only, the replication sample only, or both. It changes from study to study, even if both discovery and replication ancestries are the same. This information might be possible to obtain from the abstract of the publication, or sometimes there's a column in each file reflecting the sample size, but I'm trying to make a pipeline for many traits in parallel, so it's not really feasible to check each abstract individually. Any suggestions on how to get the sample size more efficiently?
Okay, so if you're talking about the summary statistics files, so they should always match the discovery sample information that's provided in the catalogue. So so the summary statistics always match the discovery sample, even if there is a replication sample listed in the metadata. And actually the top associations might be extracted from discovery plus replication, but the summary statistics themselves are always the discovery only.
So we have another question. It's about the raw data. Does GWAS Catalog provide synonym data for phenotype labels?
So the best way to get that information is to click through to the or search within the EFO, so the experimental factor ontology. If you go to their webpage, which you can click through from the GWAS Catalog, that should give you all the synonyms that are included for that term.
Okay, so we have another one. It's about the genome builds available for the association.
Yeah, so I think you're asking if we make the associations available on other genome builds, and we don't do that at the moment. I mean, there are tools available for liftover, but we don't provide that data within the catalogue. So they're all on the same genome build.
So thanks a lot again, Laura, and thanks everyone else for joining in.
Thanks everyone for joining.